as I briefly highlighted at the ESMO TAT conference, that 1535 has been tested and has been consistently able to show high tumor growth inhibition in a broad range of mouse PDX models as well as mouse allograft models. So those models span both the GBM tumors driven by EGFR variants 2 and 3, single nucleotide variants, and by amplified EGFR. But they also span the intrinsic and acquired resistance models that I talked about as well. Also, it's important to note that 1535 extends, as I mentioned, the survival in an intracranial PDX model too. At the ESMO TAT conference, I focused on 2 preclinical studies. The first was a mouse PDX model expressing the complex uncommon mutation. It was G719C plus S768I. So the rationale for running and highlighting that model was to show the potential to treat the intrinsic resistance mutations that I talked about that are inadequately addressed, in our opinion, to the current approved EGFR inhibitors. In that study, after 14 days of dosing, both the 15 milligrams per kilogram and the 50 milligram per kilogram arm showed high levels of growth inhibition and delayed tumor outgrowth after cessation of dosing. And importantly, there was regression observed in all the animals. There was no body weight loss during the treatment. The second preclinical model that I showed was to highlight the potential of 1535 to treat those patients that have acquired resistance to first-line, third-generation EGFR inhibitors like osimertinib. So I didn't show data, but I did mention that with respect to the acquired resistance mutations, we've demonstrated in washout studies that 1535 retains the ability to irreversibly bind to the serine residue of the C797S mutation. So complete inhibition of phosphorylation of EGFR is maintained for over 24 hours after washout. And that contrasts with osimertinib, where all of the activity was recovered within 30 minutes of washout. I showed just one of the models that we've run to support our case in this space. So 1535 in that model induced regression in, this is a mouse subcutaneous tumor model driven by the EGFR exon 19 deletion plus C797S resistance mutation. So at 40 milligrams per kilogram, BDTX-1535 induced complete regression of the tumors without body weight loss. And again, in contrast to osimertinib that was dosed at the clinically relevant level, that showed no growth inhibition and really matched the vehicle control growth. So we concluded from all these studies that 1535 has the potential to address the unmet medical need in patients with both intrinsic or acquired resistance to approved EGFR inhibitors that either have or do not have CNS metastases. And we also use the exposures from these studies across all of the models, as well as the CNS exposure that we obtained in our intracranial model to help us define the expected efficacious dose in human.